Now, it turns out that only about 2% of the DNA in your chromosomes actually ends up making proteins. So 98% of your DNA actually seems to do nothing. It doesn't really have a purpose. So we kind of make a distinction between the DNA that is going to make a protein, which we call a exon. So right here is a coding region, an exon. And then the DNA that is non-coding, that doesn't actually make proteins. We call those introns. Now, it would be kind of wasteful for the whole process of copying the DNA to make messenger RNA and then taking the messenger RNA out to the ribosome to make the proteins. If all of that happened, if 98% of the stuff that was there was non-coding, that didn't actually do anything. So our cells actually do something interesting. After transcription, when the messenger RNA is built, right here, we actually have enzymes that come in and cut out those introns. They cut the introns out and get rid of them, so that the part of the messenger RNA that is sent on to the ribosome is only the pieces that can actually make proteins. So most of the DNA that is transcribed into messenger RNA actually gets cut out and then doesn't have any effect on actually making the protein. Now, those introns may have a bit of a function to them, though. They may actually enhance some of your genes, some of the parts of the DNA that actually are coding, to get transcribed into messenger RNA or not. So there's still some research going on to figure out what exactly those non-coding regions do. Sometimes we even call them junk DNA because they seem to have no purpose. So we're starting to think that maybe they aren't quite as worthless as we used to think, but they don't actually code for proteins. One thing that we can say they are useful for, though, is when it comes to mutations. So a little bit we'll study mutations, but if 98% of your chromosome is DNA that doesn't actually make proteins, that means there's only a 2% chance that a mutation is going to affect any of your actual genes that matter. So if you've got this whole long stretch of DNA and there's a mutation that happens somewhere on there, the likelihood is it's going to happen in a portion that doesn't actually affect your genes and doesn't actually affect the proteins that are going to be made. So it's kind of like an insurance policy, having all this extra stuff that can be taken out and not actually hurt you.
So you got your lab sheet. Back on your lab tables, you're going to find this sequence. This is going to be the DNA sequence that you're going to base all of your lab data off of. So you just kind of read it in order. I've got them actually cut out back on the lab table. But if you follow along, it's going to ask you to first write down this DNA strand, then make the messenger RNA strand, and then you're going to need to use your charts to figure out what amino acids are going to be coded for from that messenger RNA strand. And then you're going to start looking at mutations. So in each case, it's going to ask you to make a mutation to the DNA strand, so that'll be this first part, and then see how that changes first the messenger RNA strand, and then the string of amino acids. This is going to take a little while. It'll take us a couple of days to do, so go ahead and get started on that, and we'll see how far you get.
Okay. One thing you have to remember about mutations. Mutations always occur on the DNA strand. Now they will make changes to then the messenger RNA and the amino acid chain, but the mutations always actually take place on the DNA strand. So when you were doing your labs, all my instructions said, change this on the DNA strand. Had a little confusion with that yesterday. But anytime you have a mutation, that's where it's going to occur. Okay? So they will affect the messenger RNA and the amino acid chain. But that's only because those things are based on the DNA. But it's the amino acid chain, the protein, that we really want to look at, okay? Because that's what is important inside your body, the type of protein that's made. So if we get a different type of protein that's made, that could cause problems. Now there's two main types of mutations. There's point mutations, and there's frame shift. But each of those types, there's some different things that can happen. So what was one type of point mutation that you guys found?
Number 1.
No. So what was number 1?
Second amino acid value instead of glutamine.
And what kind of a point mutation was that?
Substitution.
A substitution.
Come on, so all of ours are the same.
Yeah. Now, technically, on number 1, you could have slightly different amino acids there because you were randomly drawing one. But so that's one type of point mutation, is a substitution. Sometimes you also hear this called a missense. But in a substitution, you have a change of one amino acid. So, for example, what was yours, Nick? I forgot what you said.
Valine instead of glutamine.
So, you used the DNA codes for valine. It got changed, so now it's coding for glutamine. And that's going to cause a single change to the protein. So if you look at how the protein was made, there'd be one little change right there. Okay? What's another type of point mutation that you guys saw? A silent mutation. And what was the nature of the silent mutation? It didn't change anything. So in a silent mutation, there's no change to the amino acid. Okay, and that happens because of something we call redundancy. What does redundancy mean?
Something changes, but it doesn't really change.
No, that's not what redundancy. That's not what redundancy means. What's that? Repetitiveness. If I said, Tyler, you're being redundant, that means you're saying the same thing over and over again. Okay? So if you look at your chart, you'll notice there's lots of different codes that give the same amino acid. In fact, I think serine, there's 6 different codes that give the exact same amino acid. That's kind of a good thing. It's like an insurance policy. So you can have a base that changes, a mutation that changes, but it doesn't actually affect the protein. So that's what we call a silent mutation. Okay? And then what's the other type of point mutation? Nonsense. So in a nonsense, you get a change to a stop codon. And that means at that point the protein stops being built.
You're saying like when you have like methionine and then it stops right away?
Right. That would be a nonsense mutation. Now it doesn't have to stop right away. I mean, it may build half of the protein. But if you're talking about, I mean, it's just like a building, right? If I put down the first 10 blocks that I'm going to make for my foundation and I just stop there, you can't really say I have a building. So if we just put down a few amino acids and then it stops, you don't really have a protein. And so a nonsense mutation, you don't have an actual protein that's built because the thing stops way too soon. Okay? Out of those three, which one is probably going to be the worst?
Nonsense.
The nonsense, because you're not even building the whole protein there. So this is very bad. Silent mutations don't have a change. We wouldn't necessarily say they're good, but they're kind of neutral. Substitutions, yeah, that was missense. You get one change, that's still not good, so we'd say those are bad, but they're not quite as bad for the cell as a nonsense mutation. Okay? And then we have frame shift mutations. What are the two types of frame shift mutations? Deletions and insertions. Okay, and I don't think we necessarily need to, write out definitions for deletion and insertion. You guys probably understand that. But the name frame shift is very accurate because what happened to all of your amino acids after that one change?
They're moving.
Well, yeah, so the codons, where they are, change. So every single amino acid on down the line changes from that point. So we can write that here. Changes all the following amino acids. Okay, and hopefully it makes sense to you. Those are very bad too, because you have these instructions to build this protein in a certain way, and now you're building it completely different. All of the amino acids change. That protein isn't going to do anything like it was supposed to. So these are also very bad.
So all of them except 7 are bad.
Yes. Mutations generally aren't a good thing. When we start looking at evolution and natural selection, we'll talk about how sometimes they can be a good thing, but for the most part. Now, what's interesting and may seem like a bit of a paradox, most of the things that we see that come from mutations actually come, the problems that we see actually come from substitutions. Even though they're not as bad for the cell as nonsense mutations and frame shift mutations, these tend to kill the cell. So when you have a nonsense mutation where you're not even building the whole protein, or when you have a frame shift mutation where the proteins that you're building are completely different from what they're supposed to be, those generally just kill the cell off and that cell dies. Okay? So even though these are kind of worse, the things that we see like cystic fibrosis and sickle cell anemia, those kind of mutations that cause those genetic diseases didn't come from these types of mutations. They came from a substitution. In fact, sickle cell anemia, the protein that is in the red blood cells, there's only one difference in the amino acid chain, but that makes a big difference to the shape of the cell. Instead of having a nice round cell that can carry oxygen, you have that stupid sickle cell that can't carry oxygen very well. So a single change to the amino acid sequence there changed that, way back when, and that got passed on. So, like I said, it's kind of interesting that the really bad ones we don't really see the effects from just because they're so bad they kill the cells off. It's the substitutions that tend to get passed on as those bad genetic diseases. But, as we'll find out, maybe sometimes good things happen from them as well. Okay, any questions there? That's pretty much all I have as far as mutations go. So, Homework for Monday is, what is the number on that? I forget. 9.5. Okay, homework for Monday is 9.5. Don't forget to do your question of the day,
We do the back.
which is right here. Yes, you do the back as well. So do your question of the day. Do 9.5 for Monday. That's all I got for you.